Hello students, welcome to Vedantu NEET English. I am Miss Gopika, your own biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats if I am audible and visible to all of you. Yes students, are we live? All of you in the chat box, put a lot of fire emojis and let's continue our molecular basis part 3. Let me also check. Yes, we are live. Awesome. Yes. Hello. I think clearly visible, audible also. Yes. Yes, I'm audible and visible. Okay. So let's start the class. So today we are going to do molecular basis of inheritance part 3, where we have to do regulation of gene, lac operon, HGP and DNA fingerprinting. Okay. So these are the 4 topics that we are left with. Yes, audio is visible, video is audible, everything done and dusted. So let's start the class. Okay. So molecular basis of inheritance, for those students who are joining it for the first time, those students who do not know what's happening, let me tell you, this chapter has a weightage of around 40 marks in your NEET examination. That means we can predict 9 to 10 questions out of this chapter. That means the weightage will go up to 36 to 40 marks. So can you miss this chapter? No. Okay. Can you miss this chapter? Of course not. Okay. My health is fine. Thank you so much. Yes. So I think students will join slowly because it's Saturday. So all of them will be busy. Let's start students. So let's see what is regulation of gene. Like I always tell you, now my students are an example of this, my NEET students, because most of the time they are very active, but there are times when they don't want to be active. They don't want to learn, they don't want to prepare, they just want to be in that, you know, like how we tell, you know, aestivation and hibernation, like that. They want to go for a winter and a summer sleep. Yes, I think all of you are that category of students where you'll be active for 6 days in a week and Sunday you just want to relax. Now same thing happens with gene students. Why should the gene be switched on all the time? See, there are genes which are switched on all the time, which are called your household genes or your consecutive genes. That is different story. But there is a gene which will be like, I do not have to be active every time. Yeah, I can be switch off. Those are called as your luxury genes. Yes, I have mentioned this question. Yes, they have asked 10 questions. Usually 9 to 10 questions are from this topic, no matter what. Okay. So there is constitutive genes and non-constitutive genes. Now what is constitutive genes? Constitutive genes are the genes which will express themselves all the time. Okay. They are the genes that will express themselves all the time, which we also call as household genes. That means no matter what happens, they have to be switched on. Okay, they have to be switched on. Then you have the second set of genes which are called as your non-constitutive gene. Non-constitutive gene. Students, this gene is also called as your luxury gene because of the fact that they do not have to be switched on all the time. They can regulate themselves. So at what places the genes are going to regulate? Okay, all of you answer quickly. Yes, let's make the target. All of you mark your attendance, like the video and share the video with your friends, which none of you are doing, and your explanation to that is, ma'am, I do not have friends. What a nice, beautiful explanation. Okay, yes. So students, we learned about, I think I wrote. Okay, yes. Now students, when can the gene get switched on and switched off, right? Ma'am, when do the gene actually regulate themselves? That is what we are going to learn here. See, regulation of gene expression refers to a very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide. That means ultimately in your central dogma, what is going to happen? DNA is going to become mRNA, mRNA is going to become proteins. This many amino acids together we already learned in last class, where we learned about charging of tRNA, how they are going to do translation, right? So they will form many amino acids combined together with the help of peptide bonds is going to form the polypeptide chain. Yes or no? Yes, I'm definitely going to help. Just listen to me carefully. Okay. Now it can be regulated at several levels. In eukaryotes, the regulation could be exerted at in eukaryotes. Students, because as you know, in prokaryotes, there is no need for so much hangama, and at the same time, transcription and translation is going to happen simultaneously there. So there is no technical portion where there is a separation, right? Because I told you that in last class I have mentioned all of you that translation and transcription happen simultaneously in prokaryotes. Why? Because there is no RNA processing. You do not have split genes, you do not have to do splicing, you do not have to do capping and tailing, right? Yes, looking different means what way? Is it because of the bindi? Okay, yes. Yes, students. Okay, hi. Now, look here, students. In eukaryotes, the regulation could be exerted. At what levels? At transcriptional level, that means formation of your primary transcript. That means when your DNA to mRNA process, that is called transcription, during that level, gene will be like, let's just switch off. Okay, gene will be like, let's just switch off. Okay, now, processing level. This processing level means what, students? Regulation of splicing, RNA processing. That means the split gene where we have exons and introns, we will remove the introns and stick the exons together, and that is called as splicing. Okay, that is called as splicing. Clear? Now, transport of mRNA from nucleus to cytoplasm. All of you answer this. Last class I told you that after, okay, before translation, that is after you're forming your mRNA, you have to transport them from where? From nucleus to cytoplasm for translation to happen, right? For translation to happen. Got it? Yes. Okay. Yes, now transport of mRNA from nucleus to cytoplasm will be the third step where the gene is like, you know what? I should just switch off, yeah. I don't want to be switched on. Okay. Then translational level. So students, all this level is clear to you. First is going to be transcription level. That means DNA going to form mRNA is called as transcription. During that time, the gene will decide that I don't want to work here. I will just switch off. Okay. Then the second step is when we do RNA processing. That is what I taught you last time. You have split genes. That means exons are there, introns are there. Introns are the non-coding sequence. So it's not going to help us when we are forming the polypeptide chain. To remove that introns, we do a process called as splicing. During that time also, gene can decide to switch off. It's up to the gene, okay? You are no one to ask. Then the third step is, students, after we form the mRNA in the nucleus, this mRNA from the nucleus has to go all the way to the cytoplasm. Okay, it has to go to the cytoplasm. That means that what I formed inside the nucleus has to be transported into the cytoplasm. During this time also, the gene can decide to switch off. Okay, and the last process is going to be translational level. Okay, yes. Thank you. Thank you, guys. Okay, so understood, all the 4 levels are clear? Super duper clear? Thumbs up in the chat if it's clear, okay? Even if we have less students, no matter, students, if you people are active, it is more than enough, okay? I have no restriction that I have to, you know, make sure that every single student I have to beat them and bring them to class. No, whoever is there, they should learn everything thoroughly and go. Okay, and students who are watching the lecture later also should feel that satisfaction. Yes, I opened the right lecture and I'm happy. Okay, clear? Thumbs up in the chat, all of you. Perfect. Okay, this is clear. So this 4 levels question can come to you, MCQ can come, that which gene regulation happens at which level. They can give you randomly any level, so you have to be clear first if it is asked about eukaryotes or prokaryotes. They will directly ask you eukaryotes because it's mentioned here, and they can give you any of the step, they can give you any step missing, you should be in a position to answer. Okay, now we will learn about lac operon. Okay, so students, understand, gene regulation happens at 4 levels, but there are different models, operon models that will be there in our body. Okay, now luckily you are only going to study about prokaryotes, because in eukaryotes again it's going to be more complex. So in that, there are some genes are there who is going to be like, I will control you. Okay, you will release enzyme or not, I will control. So there are regulatory genes, there are promoter genes, there are operator regions, there are structural genes. So these entire thing concepts will make your operon model. Okay, so look here, the genes in the cells are expressed to perform a particular function or a set of function. For example, if an enzyme called beta galactosidase is present or is synthesized by the E. coli, it is used to catalyze the hydrolysis of disaccharide. That is, we have to break down lactose into galactose and glucose. I'm teaching very slowly, okay, so that you understand. So students, we have already learned that lac operon, there is LAC-Z, okay, which is going to release an enzyme called beta galactosidase. That beta galactosidase will break down lactose into galactose and glucose. And about whom are we talking? We are talking about E. coli here. Okay, all of you marked your attendance? Yes. Can I see all of you liking the video quickly? Yes. So students, the bacteria use them as a source of energy. Hence, if bacteria do not have lactose, listen to this carefully, if bacteria do not have lactose around, then them to be utilized for energy source, they would no longer require the synthesis of enzyme beta-galactosidase. That means I do not have lactose around me. Okay, what am I trying to say? I do not have lactose around me. Then why will I need an enzyme who break down lactose to galactose and glucose? Just think about it. Okay, you do not make any rice flour at your home. You buy it from the shop. Then why do you need a machine that is going to do the powdering? Simple, no? Common sense. I do not need that machine. I do not straighten my hair every day. Why do I need a straightener? Same thing. If the E. coli thinks that, okay, I do not have any lactose in my body, I do not have any lactose in the surrounding that I can absorb, then I do not need the enzyme beta-galactosidase. So that means it's not necessary that this enzyme has to be released every time. We can control it. Okay, we can control it. Clear? Guys, just give me 1 minute. Okay, just give me 1 minute. I think, okay. One minute, just give me 1 second. Yes. Okay, let's make the target. Is that clear to all of you? Okay, is that clear to all of you? I forgot to do one thing. Just give me 1 second. I'm just add that, okay? And guys, today is MVSAT's last day. So those who haven't written, there's no more time. So quickly go and write your MVSAT, okay? Don't wait for next Diwali to write it. Oh, ho, ho. Yes. Okay. Just give me 1 second. Okay, now I think it's not happening, students. Okay, slowly we'll do it, no problem. Okay, yes. So students, yeah, today is 26th, no? Yeah, tomorrow is the last day, 27th. Okay, 27th is the last day. So do not forget, all of you write it. Okay, all of you write it. And just see at least where you stand. Okay, your results you have to see, no? That is more important actually. If you know your results, then it is good. Right, okay, I stand here, I should prepare here, I should make this better. Okay, just 1 minute and I'm done. Yes, done. Okay, let's continue, students. See, bacteria do not have lactose around them to utilize for the energy source. They would no longer require the synthesis of the enzyme. Okay, now therefore in simple terms, it is the metabolic, physiological or environmental condition that regulate the expression of the gene. So understood? That is, regulation of gene totally depends on if there is a need, okay, if there is any physiological needs, if there is any environmental conditions. Okay, if there is any environmental conditions. Okay, clear to all of you? Super duper clear? Yes. Okay, let's continue. Now, the development and differentiation of embryo into an adult organisms are also a result of coordinated regulation of expression of several set of genes. So understood? That means when an embryo is growing, okay, when an embryo is growing, we should understand that every gene will not be active every time. There will be some genes who will take up some job, they will switch off, then there is another set of gene which will switch on to do it. So this way what happens, everything will be regulated properly. Okay, everything will be regulated properly. Very nice. Okay, now, okay students, I think I'll explain this, then we will come to GM Express. Okay, I thought I'll draw the lac operon concept first, but let me explain this. The genes in a cells are expressed to perform the same function. Oh, what happened? Okay, in prokaryotes, control of the rate of transcriptional initiation is predominant site for control of gene expression. So here we are talking about who? We are talking about your prokaryotes. Okay, so in prokaryotes, control of the rate of transcriptional initiation is predominant site for control of genes. That means transcription is the first ever place. Okay, this question has come as a PYQ. Listen to me carefully. First, where is the first place the gene can decide to switch off? Transcriptional level. So the question can come, where does the gene decide to switch off initially? What will all of your answer be? Initially, what is the answer? That is going to be your transcription. Okay, so this question is very, very important. I'm putting a mark here, and all this PDF you will get it on Vedantu NEET English Telegram. I'm so sorry, last PDF got deleted from here. I don't know what happened, who deleted, what happened, no idea. I had saved it and then I shared it also, but the textbook came instead of this document. Okay, so that document vanished. Are you all here? Yes? Can you answer me the question? Where does the DNA or where does the gene decide to switch off initially? At first level, switching off is going to happen at the transcriptional level. So in transcriptional unit, the activity of RNA polymerase at a given promoter is in turn regulated by the interaction with accessory proteins, which affects its ability to recognize the start site. Now all of you know, when we learned about transcription, we spoke about sigma factor and rho factor. Sigma factor is actually going to help the RNA polymerase tell that, you know what, you have to start initiation here. So come follow me. Right? Come follow me, we will go together. And then there is a statement in NCERT saying that RNA polymerase can do only one job, that is elongation. That means to form the mRNA exactly at the structural gene region is because of your RNA polymerase. Okay, are you all understanding this? Okay. Now, which affects its ability to recognize the start site. These regulatory proteins can act both positively and negatively. The accessibility of promoter region of prokaryotic DNA is in main cases regulated by the interaction of protein with sequence termed operator. Do not get yourself confused. All of you know PIPO-ZYA. All of you type PIPO-ZYA in the chat box. Let me see who is going to get the spelling right or wrong. Quickly. PIPO-ZYA. In that PIPO-ZYA, O is the operator region, P is the promoter region, I is the regulatory gene. That means who is going to be like, hey, you know what, you get released or not, I am going to decide. Okay, I am going to decide. Then you have ZYA, who are the structural genes. They will work depending on if this repressor went and bound with the operator or repressor went and bound with the inducer and became inactive. All of you, spelling wrong. Okay, spelling wrong. Suharshita, spelling is wrong. Madhumita, spelling is right. Evening Star, Sneha, spellings are right. Okay. Yes, PIPO-ZYA, that is PIPO-ZYA, that is your Operon model. Okay, now look here students, the operator region is adjacent to the promoter element in most operons, and in most cases the sequences of the operator binds a repressor. That means what? Repressor protein will go and bind with the operator and tell RNA polymerase, you know what, today I am not in mood to do transcription, so let's just switch off. Okay, so that is how they are going to behave. Each operon has a specific operon and specific repressor. For example, lac operon is present only in the lac operon. Wow, 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 wow. Okay, lac operator is present only in the lac operon. Okay, and it interacts specifically with the lac repressor only. So is this clear to all of you? Any doubts? We will go to lac operon. Okay, first we will write here few things. Okay, some important points we have to write. Ready? All of you? Okay, ready. Fine. Okay. So students, first we will write about two different genes. As I told you, there are two different genes in our body. Okay. The two different genes are going to be, this is GM Express, this is Gopika Ma'am Express, where I will give you a overview of what we are learning. So two types of genes are going to be there, which are called as constitutive. Okay. Why is this spelling? Suddenly I get stuck in some spellings which I cannot write it after I get stuck. Okay. Constitutive genes. Yes, I think I've got it right. And non-constitutive. Sometimes it happens, no? When we have lot of I, E, U. I'm like a normal human student, I get confused. Okay, constitutive genes, right? PIPO-ZYA, it's correct only spelling. PIPO-ZYA. Spelling is correct. Okay? Yes. Only this, some people's spellings are wrong. So look here, Students, constitutive genes, non-constitutive genes. Constitutive genes means they are always switched on. No matter what is happening, they have to be switched on, okay? Like the ones in glycolysis. That means they cannot switch off, right? Because ATP production has to happen, so it is going to be continuous. So, full time they are going to be switched on. Clear? All of you? Yes. So, we can tell that they are constantly expressing themselves. Constantly expressing themselves. Constantly expressive. We can also call them as household genes. These are the ones which will get regulated. Okay, they regulate themselves and they are called as luxury genes. Okay, luxury genes, because some days if they don't want to go to work also, they can stay at home and no one is going to ask them anything because they are rich. Okay, so same way they can regulate themselves. These are called as luxury genes. Now in this we have learned, okay, now students, two things can happen in lac operon, or let's say for any operon model. First one is going to be induction. Okay, first is going to be induction and next is going to be repression. Now what does this mean? From the words itself you understand that if we have someone to stimulate the reaction, that means here let's say we have an inducer. Okay, inducer. Now in lac operon model, the inducer is going to be lactose. So when lactose is going to be present, we are going to make sure that the repressor becomes inactive. Now in repression, what is it? Repressor is going to be there, and that repressor is not going to be stimulated or stopped by anyone. Repressor will go and bind with the operator. Is this clear? So again, this whole thing can have a negative and a positive mechanism. Positive mechanism is when the gene is switched on. Negative mechanism when the gene is switched off. And at what levels are they going to do this? They are going to start with the transcriptional level. Okay, first one is going to be at the transcriptional level. Second one is going to be at the RNA processing level. Okay, RNA processing level. Third one is going to be when they are going to transport the RNA, the mRNA from the nucleus to the cytoplasm. Okay, cytoplasm. And the last one is going to be during the translation. Okay, translation. Clear to all of you? So these four places is where the gene decides to switch on and switch off. This is clear? Any doubts here? This is just an overview I have given, okay? Just an overview. Now we will go to our lac operon concept. So let's understand what is this lac operon concept. The lac operon, the elucidation of lac operon was also a result of close association between genetist Francis Jacob. So Jacob and Monod were the people who gave the lac operon model. Okay, you can just remember Jacob and Monod even if you do not remember the full name. Okay, because question will come like that only. They were the first to elucidate a transcriptionally regulated system. In lac operon, that is here lac refers to lactose. Okay, here lac refers to whom? Lactose. Done? Yes. A polycistronic structural gene. All of you in the chat box tell me, polycistronic, is it present in prokaryotes or is it present in eukaryotes? Quickly, let's see who is the fastest finger. Tell me, is polycistronic, is it present in pro or eukaryotes? Quickly, until I drink water, I want the answer to come. Now they'll be like, ma'am, drink water slowly. Quick. Wrong. Okay, some of you are telling yes. Yeah, SM. Okay, what name is that? Yeah, guys, think properly. Think properly. Polycistronic. Polycistronic. They can make many proteins. Okay. Yes, very good. Sukanya, Madhumati, Suhashita, Roshni told different. Yes, P goes for P. Yes, perfect. I was just asking you people, but then before that all of you deleted and ran away. Okay, so every time if I tell, oh, the person has already put the correct answer, you'll all copy paste. The answer is going to be P for P. Polycistronic is going to be in prokaryotes. Okay, P for P. That means they can make multiple proteins from one gene without undergoing RNA processing. So here a polycistronic structural gene is regulated by a common promoter and regulatory gene. So students, a common promoter promoter is going to be there and regulatory genes are going to be there. Such an arrangement is very common in bacteria and it is referred to as operon. So lac operon is not the just model. Anything where arrangement is like this, that means where there is a set of promoter and regulatory gene, that is called as operon. Okay, yes, funny guys it seems. Yes, yes. Okay, all of you mark your attendance quickly. Okay, done. Now, to name few such examples, we have lac operon, TRP operon, ARA operon, His operon, Val operon. Okay. Now, the lac operon consists of very important. Okay, listen to me carefully. Lac operon consists of one regulatory gene, and what is that gene called? The I gene. Here, I is not referred to inducer, but I is referred to inhibitor. He's like, hey, you know what? I'm going to block you, right? I'm going to block you. So here, I does not mean inducer, but rather it is derived from the word inhibitor. Okay, and there are 3 structural genes. Okay, I'm going to ask you these questions later. That time, if you people confusingly answer, then it's there for you people. Okay, Z, Y, A. You're not scared enough. No, you should be scared of your biology teacher. 360 marks you're going to get for NEET. And you're not scared of your bio teacher means what, students? Okay, tell me. Yes, where is the Madhumita and Tanuja? Class readers are not sharp today. They're lazy. They're like bored, ma'am. Okay, now look here. The I gene codes for the repressor of the lac operon. So, I gene is going to form repressor mRNA. Repressor mRNA is going to form repressor protein. That protein will run and go and sit on operator and block RNA polymerase from doing transcription. That means what? Ma'am, simply gene is switched off. Now, imagine someone goes and holds the repressor. Repressor is formed. Okay, repressor protein is there. Someone goes and holds the repressor, telling that I will not let you go. Repressor will become inactive. Repressor will become inactive. Understood? Now, look here. The I gene codes for repressor of the lac operon. The Z codes for, the Z or the Z codes for beta galactosidase, which is primarily responsible for the hydrolysis of your lactose to break down lactose into galactose and glucose. Now, lactose into its same thing. Okay, now the Y gene codes for permease which will increase the permeability of the cell to beta galactosidase. Okay, it will increase the permeability so we can take in more lactose, and when we take in more lactose, that lactose will be broken down to galactose and glucose. Okay, galactose and glucose. Clear? Yes. Now the A gene, okay, the A gene encodes for transacetylase, whose function is still not known, but it is going to make the beta galactosidase more efficient. Okay, hence all the 3 gene products in the lac operon are required for the metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or the related manner. So students, this is your entire lac operon concept. Okay, look at the diagram carefully. I'm going to explain every single portion. How are you going to remember this? You're going to remember it as Pippo, okay, Pippo ZYA. Pippo ZYA is the person's name, okay, Pippo ZYA. Now, in that, what have they told? They have told that this P is going to be your promoter, okay, promoter. This is going to be the operator, okay, operator. Now, this fellow is going to be the regulatory genes, okay, regulatory gene, which is going to even control your structural genes, and these 3 together are going to form your structural gene. Clear? Structural gene. Clear? This is clear to all of you? Yes? Pippo ZYA. Some spelling and all this Rathas is putting. Okay, students, don't follow his dictionary, you will fail in exam. Okay? Yes. Now, look here. Pippo ZYA. Now, what has happened? First case, no inducer. Okay, first case, what has happened? No inducer. So, that means I is going to form repressor mRNA. This repressor mRNA is going to form repressor protein. This repressor protein will go and bind with the operator region. This repressor protein will do what? Go and bind with the operator region. Now, when it goes and binds with the operator region, just tell me that RNA polymerase, which is coming happily, dancing and coming, will it be able to move forward? Will it be able to move forward? Yes or no? Okay? Yes or no? My cyclopedia here. Yes. So what is going to happen, students? RNA polymerase, which is going to come, will not be able to cross. Why? Because the repressor has gone and bound with the operator, and ZYA is not releasing any enzymes as well. Now RNA polymerase will be like, you know what, transcription will not happen, gene got switch off. Now second scenario, okay, second scenario, what is going to happen? This is again PIPO-ZYA. I always remember that even in the second scenario, your I gene or your regulatory gene is still going to form the repressor mRNA. No doubt in that. Still it is going to form repressor mRNA. That repressor mRNA is going to form repressor protein. But the catch here is lactose is present, which is going to act as an inducer. So that means, students, a stimulant is there. Okay, we can tell a stimulant is there. Okay, how I told you induction, no? So here what is happening? A stimulant is there, which is an inducer. That inducer will go and bind with the repressor. Okay, that inducer will go and bind with the repressor and make the repressor inactive. Okay, inactive. Now imagine when sumo comes and holds you. Will all this my cyclopedia be able to sell themselves? No, they will not be able to sell because the sumo caught this person. No, not be able to move. So same way, it will become inactive. It will be like, okay, whatever, leave, life is only this much. They'll just become inactive. Same way, inducer will go and bind with the repressor and they will form a complex. That complex will make inactive. That complex will make it inactive. Now students, since this became inactive, RNA polymerase who's coming is going to happily do the transcription, and since this whole thing happened, automatically structural genes will start releasing their enzyme. What are the enzyme going to be? The Z is going to release beta galactosidase, which is going to break down lactose into galactose and glucose. And then Y is going to break down, that is, it's going to release permease, which is going to increase the permeability of the cell to take in more lactose, so beta galactosidase can break down. Then again A is going to release transacetylase, which is going to help in efficiency, to increase the efficiency. After this they will happily go for, okay, after this they will happily go for translation. Is this clear? So transcription happens, students, because see, lac mRNA is formed. Lac mRNA means what? From DNA, mRNA is formed means transcription. After mRNA is formed, this mRNA has to go to the cytoplasm. From there, translation will happen. Okay? Clear? Yes. Then life becomes zindagi, it seems. You want me to write anything? They're always ready for me to write some things. Yes. Okay, PIPO, okay, remember this portion, students. You should know which are structural genes. So these three are going to be your structural gene. Okay, these three are going to be your structural gene. This is going to be your regulatory gene. Okay, this is going to be your operator. Fine, this is going to be your operator. Done. Now what is going to happen? Automatically I gene is not going to do anything. First it is going to form the repressor mRNA. So repression can happen, no? So repressor mRNA will be formed. That repressor mRNA is going to form the repressor protein. Okay, repressor protein. That repressor protein, which is here, will go and bind with the operator. Okay, go and bind with the operator. Now that means what? Gene is going to get switched off. Okay, here the gene will get switched off. Why it's getting switched off, students? Because RNA polymerase will not be able to move forward, because here we need to form the lac mRNA, right? Transcription has to happen. So here no transcription is going to happen. So this is happening in the absence of inducer. That means absence of inducer means what? Here inducer we are talking about is lactose. Done, done. All of you in the chat, clear? Yes, Vikas Pandit, first time you are coming. Very nice to see you, child. Okay. Yes, wait, I can. Okay, done. Very nice. Now, students, second scenario, what's happening? Similar setup only. Okay, similar setup itself. Here you have PIPO-ZYA. Okay, PIPO-ZYA. This PIPO-ZYA. Okay. Okay, pipo-zy. Same thing is going to happen. Everything up to this stage is going to be same. Everything up to this stage is going to be same. Again, I gene is going to form your repressor mRNA. That repressor mRNA is going to form repressor protein. Okay, that repressor protein will be waiting to go to the operator. But here what is going to happen? Here lactose is going to be present. That means one inducer is there. Okay, one inducer is there. This inducer will go and bind with the repressor and make sure that the repressor is inactive. So, it will make the repressor inactive repressor. Okay, I am writing REP here because again place is not there. Okay, clear students? Now, what is going to happen? Here the formation of lac mRNA is going to happen. That means your transcription has happened. RNA polymerase came forward and did the process. Okay, RNA polymerase came forward and did the process. This is your RNA polymerase. Okay, it did the transcription. Now, depending on which is the ZYA, Z is going to release your beta galactosidase. Okay, beta gal. Your Y is going to release permease. Okay, and A is going to release your transacetylase. Transacetylase. Done. Now, we know that first job is going to be by your beta galactosidase. Students, it's a leaky model. That is why you will have a doubt, ma'am. Inducer is lactose. Then how again here it is happening? Okay, here it is happening. So here what is going to happen? So beta galactosidase, it is a leaky model. That means lac operon concept itself is a leaky model. Lactose has entered the system by some method in the E. coli. Okay, E. coli. Yes, ma'am ki class ki timing Tuesday, Thursday is usually my class timings, and even Saturdays. Okay, morning at 11 a.m. and 5 p.m. in the evening. Okay, so now students, beta galactosidase, what is going to happen? It is going to break down lactose to galactose plus glucose. Okay, all of you tell me this. If E. coli, okay, think you are the E. coli. Yes. No, they are not. Only this one. This is the promoter. Okay, I have written it here already. Adjacent to the operator is the promoter. I think I have written it in the diagram only. Okay. Okay. Now, students, listen here. Lactose will break down to galactose and glucose. Lactose will break down to galactose and glucose. Now, you answer this to me. If E. coli, okay, this is your E. coli. Okay, this is E. coli. You tell me this. If E. coli have glucose. If E. coli has glucose, okay, this E. coli has a lot of glucose in the body, glucose is more. Will the gene be switch off or switch on? Will the gene be switch off or switch on? Thank you, Dalton. Yes, quickly tell me in the chat box. Will the gene be switch on or switch off? This much you tell me, you will be able to understand. Okay, quick. Okay, one answer I can see. One answer I can see in the chats. Yes, very good. Ma'am, if glucose is going to be more, that means the E. coli will not need lactose. That means the gene is going to be switched off. Right? Gene is going to be switched off. But what if it is vice versa? That means glucose is very less and the E. coli cannot survive. It needs lactose. Okay, that time obviously gene is going to be switched on. Is this clear to you? Why they are requiring it? Okay, why they are requiring it? This is the answer. This is the answer. Now, lactose will get broken down to galactose and glucose. Then you have permease. Okay, permease. Permease is going to increase the permeability. Increase the permeability. Okay, increase the permeability of the cell to take up more lactose so that beta galactosidase can do the breaking. Transacetylase as of now function is not known. Okay, super duper clear. This is your lac operon model. This is your lac operon model. Okay, here some more things are there, we will just read through it. See, lactose is a substrate for the enzyme beta-galactosidase and regulates the switching on and switching off the operon. Okay, switching on and switching off of the operon. Hence, it is termed as inducer. In absence of a preferred carbon source such as a glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cell through the action of permease. Is this clear? Okay. Notes, because you will get the notes on Telegram channel, that is Vedantu NEET English Telegram channel. You can join my Telegram channel, that is Biology by Gopika. Okay, all these notes will be available there. My class timings also will be available there. Clear to all of you? Yes, fine. Okay. Now, listen carefully. Students, here, if the lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease. This is what I told you. It will increase the permeability of the cell to take up more lactose. So, imagine we have provided the E. coli to grow. Okay, in that medium, there is no glucose. But if I provide lactose, the gene is going to get switched on. Okay, gene is going to get switched on. Remember, a very low level expression of the lac operon has to be present in the cell all the time. Otherwise, lactose cannot enter the cell. That means here, what are they talking about? They telling that lactose will already, okay, a very low amount will be present, this indicating that this model is a leaky model. Any doubts in this, all of you? If you don't understand, please ask me in the chats. Okay, please ask me in the chats. Any doubts to anyone? They're telling that a very low amount of lactose should already be present. Then only inducer will be there. No, then only inducer will go and bind with the repressor. That means it's a leaky model. Okay, it's a leaky model. So, expression of lac operon has to be present in the cell all the time. Okay, all the time it has to be present in the cell, so that otherwise lactose cannot enter the cell. Okay, otherwise lactose cannot enter the cell. The lactose then induces the operon in the following manner. Okay, following manner. Clear? Now, the repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region. See, the repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing. Okay, RNA polymerase from transcribing the operon. Transcribing the operon. Yes. Leaky model means lactose has already entered into the system before it was actually supposed to enter, because beta-galactosidase have to break down the lactose, permease has to allow this entry, but before that only lactose has already entered. Okay, if the lactose does not enter initially, then lactose actual entry into the cell will not happen. Is this clear? Because that inducer is going to go and bind with the repressor. That outside stimulant is who? Lactose. Is this clear, Sukanya? So here, in the presence of an inducer such as a lactose or an allolactose, the repressor is inactivated by the interaction with the inducer. This allows RNA polymerase to access to the promoter and transcription process. So, the students, the P next to the operator, no, that is actually the promoter region where your RNA polymerase will understand, okay, I have to start from here. Okay, initiation should happen, no, that is the promoter region. That's why we are talking about that P and we are not talking about the first P. We are talking about the third P. That third P is the promoter. Okay, third P is the promoter connected with transcription. Don't blindly, be like, oh, what is this ma'am telling? Promoter connected with transcription. There you remember promoter, remember elongation, terminator, promoter, structural gene, terminator, connected with that. So this chapter is all about connection. If you connect and learn it very simple, this chapter is very easy. Okay, 40 marks will not be difficult to get. And last time also questions were very easy from this chapter. Essentially, regulation of lac operon can also be visualized as the regulation of the enzyme synthesis by its substrate. Okay, now listen here, students. Regulation of lac operon by repressor. See, regulation of lac operon by repressor is called as negative regulation. That means what are they talking about? That means when the gene is switched off. Okay, when the gene is switched off, we call it a negative regulation. That means here lactose is not there, okay, and it is not controlled by the lactose. Okay, now lac operon is under control of positive regulation as well, but it is beyond the scope of the discussion at this level. So here they are not talking to you about that because they think that you people are not into that level. Okay, yes. So I have already done so many GM Express I have put everywhere. Clear? Any doubts? Can we start HGP project? HGP. Okay. Students, we have very small portion. Okay, we'll be done in no time. Okay, so stay till the end. Do not run away thinking that okay, I'll watch it later. No, why? You have come here, finish it off and go. Okay, now HGP project, that is called as Human Genome Project. Now, see, our project or our genome is so huge that if everyone had to know about their genome, know about every single nucleotide base pairs they had, it would be complicated. Okay, it would be complicated. Okay, so here note, see, positive regulation and negative regulation is in the terms of presence and absence of lactose. Okay, that is when the gene is getting switched off and when the gene is getting switched on. Is it clear? Yes. Now, in the preceding sections, you have learned that it is the sequence of bases in the DNA that determine the genetic information of a given organism. In other words, genetic makeup of the organism or an individual lies in the DNA sequence. Agreed, all of you? I have told this before also. 99.9%, okay, 99.9% of all our DNAs are the same, which is called the bulk DNA. Then ma'am, what is different? That 0.1% is making you and me so different. Okay, so now exactly speaking, that sequence in your DNA is making you and me very different. How many times it is repeating, depending on that everything is changing, no? So here also we are talking about that. So look here, genetic makeup of an organism or an individual lies in the DNA sequence. So now you have to know the sequence. So for that we did an HGP, a mega project where many countries came together, many people were allotted telling that you will take care of this, you will take care of this. So it was a very big project, and every base pair they was charged around $3, which costed them around 9 billion. So everything was in a larger scale. Okay, so look here, if two individuals differ, then their DNA sequence, okay, should also be different. Yes or no? At least at some places. It's not necessary. Now you have fraternal twins and your the twins. Okay, so identical twins and fraternal twins. In these twins also there will be change in the sequence. It's not like everything will be ditto copy of each other, right? Sequence will be different. Now these assumptions led to the quest to find out the complete DNA sequence of human genome. With the establishment of genetic engineering technique, where it was possible to isolate and clone any piece of DNA, and availability of a simple and a fast technique for determining DNA sequence. A very ambitious project of sequencing human genome was launched in the year 1990, okay, when none of us were even born. 1990, Human Genome Project. Okay, yes, it took almost 13 years. It took 13 years. They were trying it from 1990 up to 2003. Project HG, Project HGP. Okay, yes, students, why are the likes increasing? Yeah, so sad it is for you people to like, or they're asking, YouTube is asking money, telling that one like you have to GPay this much amount. Yes, okay. Now look here. This Human Genome Project, that is HGP, was called as the Mega Project. Okay, what was it called? As the Mega Project. You can imagine the magnitude and the requirement for the project if we simply define the aim of the project as follows. See, human genome, this paragraph is very important. That first paragraph is just the introduction, but this paragraph is very important. Human genome is said to have approximately 3 into 10 to the power 9 base pair, and if the cost of sequencing required is $3 per base pair, okay, per base pair, then the estimated cost of the project would be around $9 billion US. Okay, $9 billion US. So approximately 3 into 10 to the power 9 base pairs are there, and if we have to calculate each of them, it will be $3, and a total of 9 billion would happen. Okay, 1 like is equal to $100. 1 like is equal to 1 kidney. No, like that we will start campaigning. 1 like is equal to save 1 life. So like that we will start campaigning. No. Furthermore, if obtained sequence were to be stored in a typed form, like in the sense, imagine all the sequence were typed in a book. How much? How big of a book it would be. And if each page of the book contained 1000 letters, okay, 1000 letters, and each book had 1000 pages, then each person would have 3300 such books, okay, would be required to store the information. Then what is going to happen, students? Then Ratis will keep his MyCyclopedia side and he would be carrying this book, which is around 3300 books, okay, and would be stored the information of a DNA sequence from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computer. Yes or no? If you want to type, okay, if you want to store all of the information, then what would happen? It would take so much time and so many people, so much, manpower, computer to make this fixed, right? Make this fixed. So now look here, the high speed computerization devices for the data storage and retrieval and analysis. HGP was closely associated, okay, with the rapid development of new area in biology, which is called as bioinformatics, where you are mixing biology with some computer science, okay, computer science. Clear? Yes. Okay. Now, goals of HGP. Okay, what are the goals of HGP? First of all, what are the important goals of HGP? Identifying all approximately 20 to 25,000 genes in a human DNA. That was the first target, that we will do one thing, we will identify all the genes. Okay, all the genes. What happened, guys? All of you are... Okay, yes. Yeah, thanks for reminding. I almost forget. Okay, yes. Okay, yes, clear. Now, is reflection coming? Okay, I think reflection will come. Is there any repressible operon that they are always on and switched off when needed, unlike... See, as I told you, there are two types of genes. There are genes which will be 24/7 switched on, like in the sense always switched on or completely keep expressing themselves, okay? And then there is a set of genes which are not all the time expressed, which are called as luxury genes. So under that, right now you will learn only few prokaryotes operon, okay? That is, one is lack operon. So depending on presence and absence of that inducer or that, outside stimulant, every gene can likely switch off and switch on. Clear? Okay. Now look here, identify all approximately 20 to 25,000 genes in a human DNA. Determine the sequence of 3 billion chemical base pairs that make up the human DNA. That was the main target, because if we were to do a book or if we were to type it, okay, or if we were to charge for each base pair, it would cost us around 9 billion. So the target was to make sure that we get all the sequence together. So if there is any problem, okay, any problem in the future because of one sequence or because of one set of people, okay, we notice that, okay, this one set of people have this problem, we would be able to identify, is it in the genetic makeup, is it actually in the genetic level, or is it just like that, okay, is it just like that, okay. Now, store this information in a database, okay, that was the next thing, store all this information, improve the tools for data analysis so that, we can do multiple, okay, once we understand the sequence, we'll be able to do a lot of things out of it. We can also use it for, understanding that this disorder has happened, is it because of a change in the sequence, like how we tell about sickle cell anemia, right, point mutation has happened. If I never knew the sequence, how would I know that glutamate, GUG, instead of GUG, GAG is coding? Okay, valine got coded. How would I know that there is a mutation happening? Only if I know the sequence. Okay, only if I know the sequence. Okay, clear? Yes, only some examples are given. You do not have to worry. Only lack operon you can remember. Okay, lack operon you can remember. Now, students, transfer related technologies. Okay, transfer related. Why I cannot see Tanuja and Madhumati in the class? Okay. Madhumita in the class, yes, sickle cell anemia example. See, transfer related technologies to other sectors such as industries, address the ethical, legal and social issue. Students, here you have to remember the full form of this, which is LC. Okay, since it's a girl's name, I think most of you will not forget it. LC, that full form is important. Okay, LC, sorry, okay, ethical, legal and social issues. Okay, whatever issues, okay, we will be able to address all the ethical, legal and social issues that may arise from the project as well. Okay, project as well. Now, The Human Genome Project was a 13-year project coordinated by the US Department of Energy and National Institute of Health. During the early years of HGP, the Wellcome Trust in the UK became a major partner. Other than that, we had Japan, France, China all coming together, and India also coming together for this HGP project. Okay. Now, the project was completed in 2003. Now, knowledge about the effects of DNA variation among individuals can lead to an evolutionary new ways to diagnose, treat, and someday prevent thousands of, problems, okay, thousands of issues as well. Clear to all of you? Now, methodologies. Now, this is a very, very important part. Methodologies. What were the methods they took to start the HGP project? Because students, see, I'm going to learn about sequence. Lot of sequence are there, and if I want to make sure that, okay, I understand where the problem lies, what exactly is happening, I have to start from the scratch, right? So now they took two approaches, two methodologies. First one, see, the methods involved in two major approaches was one approach focused on identifying all the genes that are expressed as RNA. That means what? All the genes who can express themselves as RNA were taken into consideration. That was the first step, and they were called as expressed sequence tags. You are expressing, I will take you in. You can express yourself as mRNA, I am taking you in, and I will label it as expressed sequence tags. Okay. Now, the other took the blind approach. Okay, they did not think about sequencing or anything. See, the blind approach of simply sequencing the whole set of genome that contained both the coding and the non-coding sequence. Okay. Now, students, you tell me, I'm taking all the things. I'm taking NEET aspirants, I'm taking board aspirants. What's the use? I'm taking a class where I'm telling that, you know what, anyone can join. Okay, you're not even from a science background, you come and join. What is the use? Will it is it going to benefit that child? It's not going to benefit. So, I will only pick a student who is a NEET aspirant or who is going to write their boards, right? So, here the second approach was an approach where they took all the coding and non-coding. Clear to all of you? All coding and non-coding sequence, and later assigning different regions in the sequence with functions, a term called as sequence annotation. Okay, sequence annotation. So, the first methodology was express sequence tags. Okay, expressed sequence tags, where they took only the set of genes which will be able to express themselves as RNA. Okay, which is again through your central dogma, if you remember, DNA to mRNA. Okay, that structural gene region which can form mRNA, right? Same way here also, only the genes who are going to express themselves as RNA, we will take it and we will call it as express sequence tags. Second approach was blind approach. Who will do categorization? I'll take all the genome, be it coding, be it non-coding, I'm going to take all of them. Then I am going to categorize them and give them each of them one function. Okay, so that was called as sequence annotation. For sequencing, the total DNA from a cell is isolated, converted into random fragments of relatively smaller size, okay, and cloned in a suitable host using a specialized vector. The cloning results into amplification of each pieces of DNA fragments, so subsequently could be sequenced with yeast. Don't worry, I'm going to teach you this. The commonly used host were bacteria and yeast. Okay, this is important. Commonly used host were who? Bacteria and yeast, and the vectors we used were BAC and YAC. Okay, BAC and YAC. Clear? Now I'm going to explain this. Okay, I'm going to explain this to all of you. Okay, so what did they just say? Okay, listen to me carefully, you will be able to understand it very, very smoothly. See students, two approaches. Okay, we are talking about the methodologies. Okay, two methodologies to approach this. First one was, okay, where they took only the genes, okay, they took the genes that will express, okay, genes that would express themselves to form RNA. Okay, I think if I write like this, you might get confused. Okay, I'll write the whole statement. Okay, genes that expressed to form RNA. Clear? This is the first method, and that method was called as expressed sequence tags called as ESTs. Okay, ESTs. Clear? Yes. Okay. What is this? Weather forecaster in the chat box? Weather forecast in the chat box, I guess. Okay. Next one. Okay. Next one is going to be a second methodology. Okay. Second approach. What is the second approach? All of you in the chat. First approach you understood. Okay. Here it was a blind approach. That means we did not categorize anything. Okay. It was a blind approach of taking all, okay, taking all coding plus non-coding, okay, coding plus non-coding sequence, and later categorize them and give them the function, okay. This technique was called as sequence annotation. What were they called, students? Sequence annotation. Sequence annotation. Clear? Two techniques are clear? Okay. Yes. Okay. Now, what is the next thing they told? How did this whole process happen? So, I will give you a very gist about it. Let's say, students, this is your whole genome. Okay. This is your genomic DNA. Now, from this, we are going to form fragments, okay. After writing this, you have to see if you understood the statement, okay. We are going to make fragments. These fragments you are going to add into vectors. Okay, these are your vectors. You are going to add these fragments into the vector. Remember, biotechnology, we have learned this. Okay, with the help of a vector, you will be able to enter into a host. Okay, then from here to a host. Okay, clear? This is what they told. Now they are telling that the most commonly used host, okay, most commonly used host is yeast and bacteria. Okay. And commonly used vector is what? To carry my desired gene, right? And the most commonly used vector is going to be my BAC and YAC. Okay, my BAC and YAC. Okay, BAC is going to be your bacterial artificial chromosome, YAC is going to be yeast artificial chromosome. Is this clear to all of you? Whole thing is clear? Yes? Clear? Check once again. Okay, check once again. Anyone has a doubt? Please ask me right now. Yes, we can amplify this. Okay, we can amplify this. So here you can add one more step where we do amplification because it's mentioned there. Okay, that will happen with the help of PCR. Now I have taught you PCR in your biotechnology, all of you remember, where polymerase chain reaction, where we can make million copies from a small piece, right? Amplification can happen. Again, we can sequence it with a sequencer. Okay, Sanger sequence will use it. Now this much is clear? Yes. Yes, yak. Okay. Clear to all of you? Chalo. This is done. Okay, now we will go back to the paragraph that we were learning and you tell me if you understood this last statements. Okay. See students, here, from here. For sequencing, the total DNA from a cell is isolated, converted into random fragments to relatively smaller size. Recall, DNA is a very long polymer and you cut them, okay, and it is cloned into a suitable host using a specialized vector. So remember, I drew a vector into that so that with the help of a vector we will be able to enter into the host. Now the cloning resulted in amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. So we will always make millions of copies, so it is easy for us to sequence it, right? Now this is clear now with that this last step. Okay, I explained it here. This is the whole step. Okay, genomic DNA, making small fragments, we will insert these fragments into the host with the help of a vector. We can amplify it with the help of a PCR, so we can easily sequence it. We will make millions of copies instead of working on a short fragment. Okay, this two approaches also is clear, I hope. Now we will go to the next part. See here, see here. Now the fragments were sequenced using automated DNA sequencer that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for developing method for determination of amino acid sequencing. So Sanger got recognition for his Sanger sequence, that is for sequencing. Second recognition he got for sequencing the amino acids to form the protein chain. Okay, now these sequences were then arranged based on some overlapping regions. Students, actually overlapping regions are too complicated to understand, so just remember, overlapping means what? One over the other. Okay, so depending on the overlapping regions, we cannot physically remove them, so we had to do it with the help of technology. Okay, so here this required generation of overlapping fragments for sequencing. Alignment of these sequences was not humanly possible. Okay, alignment of these sequence was not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. So here what is happening? Let's say Sneha is there, Sukanya is there, Madhumati is there, Newton is there. So now I am telling all of you one assignment. I am telling Sneha, you make sure that the class is silent. Okay, there are no spammers, everyone are talking, all sensible things. Okay, then I am telling Roshni, make sure that everyone is putting fire emojis when I am entering into the class. And let's say I am telling Newton, just let me know if I have updated all the notes. If any notes are missing, please update to me. Madhumati is checking the attendance, how many likes have come and how many students are going to comment. So that means what? I have annotated, I have picked all of you, then I have given all of you one function. Same thing is happening here, students. There, see, after we get the sequence done, these annotated were assigned to some. So these sequences were subsequently annotated and were assigned to each chromosomes. Clear? Now the sequence of the chromosome 1 was completed only by May 2006. So even though the whole project got over in 2003, the last chromosome, okay, which is very ironical because the name is chromosome 1, but the last chromosome to be sequenced was your chromosome 1. This question has come as an MCQ, including the date they are going to ask you. Okay, in May 2006, this was the last 24th chromosome, that is here. The sequence of chromosome 1 happened. So in this question is a PYQ, so remember this properly, PYQ. What is the PYQ? That May, okay, May 2006 we formed or we got assigned the chromosome 1 sequence. Okay, chromosome 1 sequence. Okay, now then see 22 autosomes and X and Y to be your sex chromosomes. Yes, it took time because obviously they have to sequence it, they have to know if this fellow has some other characteristic or anything else will be done, so that is why it took time. Clear? Yes, May 2006, very recent, right? Very recent. You guys are born, no? I think, are you, like, the date is not that year, only the month and the year is asked. Okay, month and the year is asked. Yes. Done. All of you in the chats, very nice, okay, very nice. Okay, now students, this were the features. Okay, what happened? See, the human genome contains 3167.7 billion base pairs. Let me tell you, your HGB project as a whole, okay, you people are born in that year, okay, 2006. So students, HGB project as a whole, so many things you have to remember. Okay, understanding part is there. See, like that whole genome thing you can understand, but some portions you have to remember it. Okay, you have to mug up a little portions. I like, see, many people will tell don't mug up, but see, all this, how much ever I explain, you will not remember. You have to learn some numbers as it is. Okay, so here, okay, now look here, the average gene consists of 3000 bases, but sizes vary greatly with the largest known. This is important. Okay, your dystrophin at 2.4 billion base pairs. So the largest known human genome is going to be dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than the previous estimate of 80 to 1,40,000. So before this HGB project, everyone thought that 80 to 1,40,000 genes are there who is going to express themselves, but when they actually checked, it was only 30,000. Okay, less than 30,000. Clear? That is 99.9% nucleotide bases are exactly same in all people. So students, technically we got to know about our bulk DNA. That is 99.9%. You and me, all of us have bulk DNA. We got to know only because of our HGB project. Clear? DNA fingerprinting also became easy because of all this only. But again, DNA fingerprinting was done in 1985, which is like long ago. But then you understood, no? All this ideation came into people that, okay, this is what is happening. Now, the functions are unknown for over 50% of the discovered genes. Less than, this question also has come. Less than 2% of the genome codes for protein. So this is also an important PYQ. This question I saw today itself. Okay, they'll ask you more than 2%, less than 2%, equal to 2%, more than 5%. So your answer should be less than 2%. Okay, less than 2% you answer. Please don't answer 2%. Okay, did you find the rest of it? No, less than 2% only you answer. Repeated sequence make up the large portion of a human genome. Now we are going to study this in DNA fingerprinting. Okay, repetitive sequences are stretches of DNA sequence that are repeated many times, sometimes in 100 to 1000 times. They are thought to have no direct coding function, but they shed light on chromosome structure. All this you don't worry, just understand that we have repetitive DNA, repetitive strands of our DNA. I will tell you about that when I am teaching you DNA fingerprinting. Chromosome 1 has most genes, that is around 2968, and Y gene has around fewest, 231 genes. 231 genes. Okay, this is 968, like a phone number, no, 968. Okay, and this is going to be 231. Scientists have identified about 1.4 million locations where single base differences, that is SNPs, are present. Single nucleotide polymorphism is present. Can I get 3 people's name from the chat? 3 people's name. Okay, 3 people who are ready to sacrifice their names for me can give me your names quickly. Fast. Let's see who is going to sacrifice their name. Guys, we'll take a 5 minutes break and then we'll start DNA fingerprinting, because I want all of you to, just take that 5 minutes break. Just water break, nothing like long break or anything, just 5 minutes, okay? So that all of you can go refresh and come back. Because DNA fingerprinting, I want all of your concentration very, very clearly. Okay, we have Technoblade, Charan and Sneha. Okay, Technoblade, Charan and Sneha. So we are just going to write T. Okay, Technoblade T, Charan C and S for Sneha. Now let's say, students, we are going to see 3 people's DNA. Okay, one small repetitive strand we are going to take. Okay, this is one strand. Now, if you look at this, if you see a Technoblade, Charan and Sneha's, you will notice that at one position, all the 3 have different nucleotides. Yes or no? See, Technoblade has AGAT, Charan has AGCT, Sneha has AGTT. Now, if you see, it is just one. Okay, this one change, students, that can make a lot of difference, because in sickle cell anemia, we just saw that instead of GUG glutamate, it became GAG, which coded for valine. This caused sickle cell anemia, which is a deadly disease. So don't you think one nucleotide change can affect a lot of it? So this is called as SNPs. That means single nucleotide polymorphism. So here you can say which occurs in humans. This information, see, scientists have identified about 1.4 million locations where single base DNA difference, that is one base, just one base has changed. okay, because of which Technoblade would have had a different, you know, completely different thing. Charan would have different, Sneha would have different. It could be even with your hair texture. Now let's say all the three of us, okay, all these three people have straight hair, but Technoblade has a more frizzy hair, Charan has a more soft hair, this Sneha has a more like wavy hair. Okay, all of them have a straight hair, but the end have some curls, or some people's are brown, some people's are black, texture is different. Okay, so like that. Small minute changes in our bases can lead to so much differences. Okay, example is again your sickle cell anemia. Is this clear to you, all of you? Single nucleotide polymorphism, SNPs full form they can ask. Okay, SNPs full form they can ask. Students, next topic is going to be DNA fingerprinting. So before that, okay, let's take a 5 minutes break. 5 minutes break, DNA fingerprinting, then we are done. Okay, then we are done for the day. Okay, yes. So this Diwali, she is coming. I do not know who is coming, but something super duper exciting is coming for all of you. Since Diwali season is very special in Vedantu, you are going to get something amazing. So she is coming, and when is she coming? So I got to know, okay, she told me, she called and told me that I'm coming. Usually she will not tell, but yes, I requested her, telling that my GMAs are going to question me, so I need the answer. So she told me that she is coming on Monday evening. She's coming on when? Monday evening. Don't tell it to everyone else, okay, then she will not come. Okay, she will not come. Monday evening she is going to come. So wait for her to come. Okay, wait for her to come. Clear to all of you? Yes or no? Clear to all of you? Yes? Yes. Done? Yeah. So Monday evening she is going to come. So wait for her to come, and by then take a cutie break and also come. Okay. Okay, 5:20 already. So we will come back by 5:25. Okay, 5:25. 5 minutes break is enough? Okay, 5 minutes break is enough? Yeah, new zoo teacher, because you people are not liking my video, no? So I thought, okay, let she come. Okay. Fine, happy. Introduction of new zoo teacher. She's coming on Monday evening, okay? Then that day you people will give me farewell or what? All joke. Diwali something is coming, okay? Wait, stay tuned. Monday evening is the thing. Don't tell anyone that Gopika Ma'am told. It's difficult, very difficult. I got her phone number and I asked her. Okay? Okay, come back after the break. Okay, ready, steady. Let me tell all the lazy people. Okay, yes, let them join. Okay, so she's coming Monday. The video is already live, you can see, and you will get to know what is happening. Okay, she's coming, who is coming, all of that. Let's do DNA fingerprinting. Ready, all of you? Okay, chalo. Yes, let's start. Yes, we are back. Okay. So students, DNA fingerprinting, as stated in the preceding session, 99.9% of the base sequence among humans is the same. So that means in Technoblade, Charan and Sneha, 99.9% also was same. But that 0.1%, that is the most important part. Okay, assuming human genome has 3 into 10 to the power 9 base pairs, and many base sequences would be their difference. Okay, it is these differences in the sequence of DNA which make every individual unique. Okay, in their phenotypic appearance. If one aims to find out the genetic difference between two individuals or among individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare two sets of 3 into 10 to the power 9. Let's take example, Deekshi is there and Sony is there. Imagine I have to compare between these two people. Every single time I have to do 3 into 10 to the power 9 base pairs for these two people. It will be such expensive process. Okay, so imagine comparing two sets. DNA fingerprinting is very quick way to compare DNA sequence of any two individuals. So students, exactly what is happening here. This was done by Alec Jeffrey. Okay, Alec Jeffrey was the person who found out DNA profiling or DNA fingerprinting. Okay, what's his name? Alec Jeffrey. Okay, in 1985, he worked on DNA fingerprinting. Now here, read this, then I'll explain. Okay, first we will read this. See, DNA fingerprinting involves identifying differences in some specific regions in the DNA sequence called as repetitive DNA. So they're telling that we have to find differences in the sequence at one spot, which is called as your repetitive DNA, because in these sequences a small stretch of DNA will be repeated many times. So here he's talking about, or they're talking about, a small stretch means what? Let's say AGAC. This small stretch will be repeating multiple times. Okay, these repetitive DNAs are separated from the bulk genomic DNA. Okay, that means your 99.9% is going to be your bulk DNA. Okay, is different peaks during the density gradient centrifugation. Again, I'll explain all of this, just read. The bulk DNA forms a major peak and the other small peaks are referred to as the satellite DNA. That repetitive DNA, okay, that repetitive DNA, we will call that region as the satellite DNA. Depending on the base composition, A and TC, okay, depending on three things, we can categorize the satellite DNA. First is depending on the base composition. If that composition has AT rich region or GC rich region. Second thing is depending on the length of the segment. And the third thing is number of repetitive units. Now, the satellite DNA is classified into two major categories, which is called as microsatellite and mini satellite. Okay, these sequences normally do not code for any proteins, but they form a large portion of the human genome. So this is the reason why we tell less than 2% genes code for the protein. Even though these genes are not coding for the protein, they are going to contribute the major chunk of a DNA. Okay, major chunk of a DNA. Okay, now these sequences normally do not code for any protein, but they form a large portion of the human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, anything in the crime sequence, students, now all of you will become, all of you are going to be CBI. Okay, imagine crime scene happened next to your home. So one crime scene happened where there was a jewelry, okay, let's say one Spanish, okay, some queen, okay, Queen Victoria's chain got lost, okay, it was kept for an exhibition in the museum and that chain got lost. So during that, you know, stealing process, the theft, what happened, some blood sample we got, okay, some blood sample we got. That blood sample, with that blood sample we are going to find out who was present in the crime scene, okay. Now when I am doing that, I am not going to bother about the 99.9% of the bulk DNA. What is going to make you and me different from that person? 0.1% DNA. Clear? 0.1% DNA. Now, since DNA from this every tissue, it could be what we got blood sample. It is not necessary that every time we get blood sample. It could be one hair follicle, okay. It could be a hair, it could be nails, it could be saliva, it could be any of this, because every time that person will be like, oh wait, let me put little blood and go for the forensic. It's not like that, no. Sometimes we get different things. Now imagine a murder happened and during that the girl, okay, let's say the girl got murdered and then there was a boy or another girl. Imagine she pulled that person's hair and by luck between her nails we got the hair follicle. Okay, we have to get it from the follicle, okay, hair follicle. With that follicle, with that tiny bit of a follicle, we will be able to do the DNA sample. Okay, now from an individual show the same degree of polymorphism, they become very useful identification tools in forensic application. Furthermore, as polymorphism are inheritable from parent to children, DNA fingerprinting is based on the paternal testing as well. So now students, very important is your paternity test. That means in abroad and all, most usually it happens. Here and all it will not happen. They'll give one on your head and they'll be like, you're my child. Okay, but in foreign countries, the child or the parents, they cannot force, they cannot, like, you know, hit each other. All that is there. No, too many conditions are there. So the child will tell, I don't think this is my parent because they do not take care of me. I feel I'm adopted. I need to do a paternity test. That means what? We know that 50% comes from our mother, 50% comes from our father. By looking at both the parents, we will be able to understand who the child is. Right? Let's say we got the child DNA. From the child DNA, if I compare parent A, parent B, I will be able to understand that half, 50-50%. Okay, 50-50% is going to form the child's DNA. We understand, okay, this is the parent of the child. But in India, we can't do that because they'll give you one on your head and you'll be like, you're lucky that you're born to me, they will tell. Yes or no, students? Tell me. Yes. Correct. Do you agree or not? If you tell your parents that I feel that I have been adopted because you're not taking care enough of me, next minute, they'll be like, if I would have adopted you, I would have adopted a better piece. Right? I would have adopted a better person itself. Okay, why would I adopt you? So this is a common thing in India. It's a very funny thing, no? How our parents talk, and they're so lovely that they talk like this very casually. Very casually they tell such thing, but this cannot happen in abroad. Okay, if a parent would tell something like this, that child can go and give a case and everything and all that. Here, nothing like that. Okay, so yeah. So as polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting, it is essential that we understand what is DNA polymorphism. Means in a simple terms, okay, polymorphism means variation at genetic level. Okay, variation at genetic level arises due to mutation. Okay, arises due to mutation. Now, recall different kind of mutation and their effects that you have already studied in chapter 4 and in preceding sessions in this chapter. New mutation may arise in an individual. Okay, new mutation can arise in an individual either in the somatic cell or in the germ cells. Cells that generate gametes. So germ cells means the cells that form your gametic cells. Okay, somatic cells are the other cells. Okay, generate a sexual reproduction organ. If germ cell mutation does not seriously impair the individual's ability to have an offspring who can transmit the mutation, it can spread to other members of the population through sexual reproduction. That means in case in the germ cell mutation has happened and if that, okay, does not lead to a problem of, you know, having kids, then it is fine, but it can definitely get transferred because you know that every generation, every character will not be dominant. Sometimes they'll become recessive. After 2-3 generations, they'll become dominant. Right? You have studied in principles of inheritance. Now, allelic, again, recall the definition of alleles from chapter 4, sequence variation, has traditionally been described as a DNA polymorphism if more than one variant at a locus occurs in a human population with a frequency greater than 0.01. So students, all this I am going to explain it to all of you. Okay, just listen to me carefully now. Okay, let's okay, see what is your DNA fingerprinting. Okay, listen to me 5 minutes carefully and then you will understand everything. Okay, yes, Pradeep Kumar, Sony, Mike, everyone listen. Okay, next 5 minutes is very important for your life because you will understand something. Okay, now students, let's say this is the, okay, this is our Queen Victoria's chain, okay, made up of precious something. Okay, this was there. Now imagine from this place I got a blood sample. I got what? I got a blood sample. It need not be always a blood sample, it could be saliva, it could be hair follicle, anything. Now what is my job? From this blood sample I need to find, okay, I need to find the DNA. Now they are telling me that DNA, okay, so this crime scene is there. This is our crime scene number 1. Okay, I have a nice question for all of you, so you should answer that question later to me. Okay, so this is the crime, students. Let's keep the crime scene there. Okay, we will wait. Now before that, let's understand about the DNA. So they are telling that DNA is your genetic material. That one, in the DNA 99.9% is same for all. Okay, is same for all, and I will call this as the bulk DNA. In some books you will see it as junk DNA also. Okay, junk DNA or bulk DNA. Now we will keep the word bulk DNA. Then they are telling that 0.01%, okay, is going to be called my repetitive DNA. Okay, repetitive DNA. That means let's say again I want 3 examples. Okay, 3 people. Who are the 3 people I can take? Crime is this: Queen Victoria's chain got stolen. Okay, my God, I hope no one comes and catches me, yeah, for telling such things. Okay, 3 people. Let's say, okay, by the time you people write your names. Now students, they are telling that this repetitive DNA, okay, they can have repetitive sequence. What is repetitive DNA? A small sequence getting repeated. Let's say this is the sequence, okay, AGAC. This is the sequence. Now another person, let's say, who told a Technoblade, I already took your name, no? Sony, Sukanya and Madhumati. Okay, Sony, Sukanya and Madhu. Now let's say students, this AGAC, it may not be necessary that all the 3 will have it. Now let's say this is Sukanya sequence. Okay, Sukanya sequence. Now Madhumita will have AAC. Okay, Madhumita will have this and Sony will have AGAC. So now Sukanya and Sony has the same sequence. Ma'am, does that mean that they're twins or something? No. Now it is, what is going to happen, students? One scenario is that like Madhumita and Sukanya, different repetitive sequence can happen. But imagine both of them have the same sequence. Hi Bhavana, you were in the KCET channel, no? I remember. So students, imagine Sony and Sukanya has the same sequence. Now what is going to happen in that? Let's say this sequence in Sukanya is repeating 10,000 times. 10,000 times the same sequence is repeating. It could be continuous or it could be with break. Okay. Now in Sony, the same sequence is only repeating 100 times. 100 times. You understood? The repetitive sequence can be repeated, okay, different times in different people. See, 10,000 times in Sukanya, 100 times in Sony. Does not make them similar at all, right? But at the same time, you see here, different repetitive sequence. It can repeat how many ever times in Madhumita. Clear to all of you? Clear to all of you? Is this clear? You understand this clearly? It's very important. Now, they are telling the sequences that we are talking about can differ. The satellite regions that we are talking about, the repetitive sequence, that region we call it as a satellite region. Okay, the region we call it as a satellite. Okay, satellite region. They are telling that the satellite region can depend on 3 categories. Okay, depending on the base composition. Okay, depending on the base composition. Okay, depending on number of repetitive sequence. Okay, number of repetitive sequence. Now students, all of you are studying loci, okay, we tell no on the gene. So this one, okay, this will be one in one loci, okay. Now listen here, depending on the base composition, because depending on the number of repetitive sequence and the length, okay, and the length of the sequence, we can categorize this microsatellite and this satellite regions, okay. We can categorize them into two categories. What are the two categories? Mini satellite regions and microsatellite regions. What is it, students? Mini satellite regions and microsatellite regions. Depending on what did I categorize them? I categorize them depending on this. So far good? Anyone has a doubt? Please let me know in the chats quickly. Is this clear? Fast, 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 fast. Okay, fine. Now this mini satellite region, students, this region is rich in A and T region. Okay, it is rich in what? G and T region. G and T region, it is rich. Clear? And the length will be around 10 to 10,000 base pairs. Okay, 10 to 10,000 base pairs, and it is can repeat around 500 to I think 10,000, 1 lakh times. Okay, 500 to 30,000 times. 500 to 500, yeah, 500 to 30,000 times. You do not have to remember this fully. Just remember it is GT rich region. Okay, microsatellite region is AG rich region. Okay, 2 to 6 base pairs. 2 to 6 base pairs. Okay, this is your microsatellite region. Now, understand that for every person, that's 0.1%, this one. Okay, as I spoke here in this Sukanya's, Madhumita's and Sony's case, students remember it could be, see here, if it is a microsatellite, it could be like this. Okay, AGA region. Okay, this one repetitive sequence will repeat around 100 to 10,000 times. Okay, here let's say this is a different sequence where it is more longer. So all this you don't worry, just understand microsatellite, mini satellite region. Mini satellites is GT rich region, that means guanosine, thymine rich region, and your wait, did I write it wrong? GC, no, I wrote it T. Sorry, GC rich region. I'm telling C, but I'm writing T. Okay, and this is AT rich region. AT rich region, and this is GC rich region. Okay, guanine and cytosine rich region and adenine and thymine rich region. Clear? Yes. Okay, AT and GC. Students, how can it be AG? Okay, I wrote by mistakenly, you people continuing there. AT is adenine and thymine, no, they both only will bond. GC is going to be guanine and cytosine. Okay, so this is your two regions of the satellite. Now, whenever a crime scene, let's come back to our crime scene. Now, what am I supposed to do with this crime scene? I'm going to take the blood sample. I'm going to extract the DNA out of this. Yes or no? I'm going to extract the DNA out of this. Now, with the help of restriction endonuclease, okay, I extracted the DNA. With the help of restriction endonuclease, I'm going to cut the DNA. Okay, I'm going to make it into smaller fragments. After that, I'm going to run it in an agarose gel electrophoresis. So, depending on the size, I will get the fragments. So far, good? Clear? This is clear? Okay. So, that and all is not mentioned very well in your NCERT. That is why most of the time students get confused. Now, let's see what you would do. Okay, now say Victoria's chain has gone. Okay, Queen Victoria's chain has gone, and you people are the CBI. Police people has transferred the case to all of you. Okay, now what you will do? Your biggest luck is that you got blood sample from that crime scene. Okay, so first you will isolate the DNA from that crime scene. Okay, from the blood sample that you got. Okay, from the blood sample you got, you will isolate the DNA. Okay, you will isolate the DNA. Then you will digest the DNA, not like eat it, okay, like I mean you will cut the DNA with the help of restriction endonuclease. Okay, restriction endonuclease. Yes, now separation of DNA fragments by, so after that students, with the help of a gel electrophoresis. Remember all of you gel electrophoresis? In biotechnology we had studied different wells will be there. In these wells we will add our sample and depending on the size, okay, depending on the size it is going to run, okay, depending on the size it is going to run, correct? Yes, so it will run. Now look here students, till then I told you, in gel electrophoresis we cannot see it. On the agarose gel you cannot see till where the sample run. So after that you will transfer it to a blotting. You will transfer it to a blotting that is called as southern blotting. That means with the help of a nitrocellulose and nylon sheet you will transfer it. That means this nylon and nitrocellulose will absorb the strands, okay, it will absorb the strands. So after that, okay, after that you will use something called as a VNTR probe. VNTR probe, let's say, okay, so guys, I'm going to take your name as the criminals, okay, is it okay for you people? If anyone doesn't want their name to be used as a criminal, you can withdraw your names. Yes, can I use 𝐂𝐤, Madhumita and Soni's name? So you three are the crime people, okay, criminals in that scenario. Now among that, who wants to be the criminal? Raise your hands. So students, now if I go back here, okay, one second. If I go back here, these are the sequence I got, right? AGAC, GAAC. Now these three are the criminals. Now when I'm making a VNTR probe, okay, from the crime scene I got the blood sample. I already have the blood sample. Now I know which is the sequence that is repeating. Okay, now here I take three suspects. These three are my suspects. Okay, these three suspects. Out of these three suspects, one person is going to be the criminal, right? One person is going to be the criminal. Now all of you want to be criminal. Huh? Shame on you people. How can you, ma'am, voluntarily I'll be the criminal. Now students, I know that this sequence, okay, let's say this sequence is repeating in the crime scene blood that I got. Okay, how much it's repeating I don't know yet. So I will make a VNTR probe, that is A for T, G for C, A for T and C for G. This is complementary. VNTR probe means complementary strand. Okay, complementary strand. So out of 𝐂𝐤 and Soni, one of them can become the criminal. Right, because I know that this is the sequence repeating. I have made a complementary strand for that. Now this VNTR probe will go and exactly bind there. Okay, it will go and exactly bind there. Clear to all of you? So hybridization using labeled. So this is a radioactive material. This is a radioactive material. That means when I see it under the auto radiography, I will be able to notice that one thing is like this. Okay, it is radioactively labeled. Okay, it will stand out. Yes. Now, detection of hybridized DNA fragments by auto radiography. A schematic representation is shown in below. So look here. Sorry, this one. Look here, students. Now this is the schematic representation. So let's say this is what we got. Okay, these are the chromosomes number. We are only let's say we are only thinking about one chromosome. So students, look here. You have paternal chromosome, maternal chromosome in both the sides. Okay, individual A. Okay, so let's say this is 𝐂𝐤. Okay, this is 𝐂𝐤 and this is Soni. Okay, this is Soni. Okay, these two people are there in the crime scene and we got a blood sample also. We got one blood sample. Now, if you look there, you will know that different numbers are there. Okay, different numbers are there. Now, when you run it on a gel electrophoresis, you will be able to see the fragments. Understood this? No, you will be able to see the fragments. Now, students, you tell me who is the criminal? A or B or I mean, this is the sample I got from the crime scene. Okay, this is the blood sample I got from the crime scene. Now, you tell me who is the criminal? Quickly in the chat, who is the criminal? Yes, Priya is telling B is the criminal. Others, what do you others think? Quickly. Soni is the criminal? Yes. So here we will understand that this C sample and B sample, you can see the same fragments. Yes or no? same place, okay, in the gel electrophoresis, same level, the fragments have gone and stopped. So here Sony becomes the criminal. Now students, you tell me answer for this. So here this is a little more complicated one. Try to look carefully and understand. So this child got kidnapped, and the child is missing, and from the kidnap place we got some blood sample. Okay, we got some blood sample. So from the blood sample we have to identify who is the parents. Okay, who is the parents? Is it A and B, C and D, E and F, or G and H? Who is the parent? So this child is missing, we got some blood sample. Now we have to identify the parents. So we have taken 4 parents who have come and told that my child is missing. Okay, 4 parents came and told my child is missing. So from all the 4 parents, okay, we have taken blood samples and we have done a gel electro, we have done a DNA fingerprinting, and these are the fragments that we have got. Now remember that when you are doing such kind of DNA fingerprinting, all of you are CBI people, don't chumma tell answer. Okay, all of you should understand that 50% comes from the father, 50% comes from the mother. Looking at the sample, you tell me which is the parent. All of you, take your 2 minutes and tell me the answer. All of you tell me which is the parent. Students, since I drew, there is a lot of human error, but otherwise you can look and tell. Okay. Yes, quickly tell me which is the parent. Okay. See the child strands DNA you can see. Now you have to see from mother and father, if the child has got 50-50%, what is going to be the answer. Priya Prasad Warrior is telling one thing. Other people? C and D. Okay, all of you are agreeing to that. C and D is the parent. Yes, very good. C and D is the parent. Why C and D is the parent? Because if you see here, 50% has come from the father, 50% has come from the mother. See, this whole portion, okay, is exactly same here. Okay, and if you see here, this portion and this portion from the parent. Okay, this portion and this portion from the parent. So can you see, C and D is the parent. Clear to all of you? C and D is the parent. So here we got the answer that C and D is the parent. So here if you see this portion, okay, and this portion, okay, this portion is the same. Okay, same way this portion, okay, and this portion. Clear to all of you? How you understood? Yes, this is your DNA fingerprinting. So students, all the steps of DNA fingerprinting has to be pakka clear to all of you. Remember that these are the steps. This is your blood sample you got, okay, the blood sample. From the blood sample you will extract the DNA, okay, DNA will be extracted. Question will come from this only, okay, entire DNA will be extracted. Then from that DNA you are going to cut the DNA, okay, you are going to cut the DNA by using molecular scissors. That is going to be who? Your restriction endonuclease, okay, restriction endonuclease. So with the help of restriction endonuclease, we are going to cut the fragments. Okay, after that what are we going to do? After that we are going to run a gel electrophoresis, right? We are going to run a gel electrophoresis. In the gel electrophoresis you are going to have different wells, okay, where we will put the sample. After putting the sample we are going to get some rays, okay, we are going to get some fragments. Okay, like this we will get some fragments. Okay, at different levels we are going to get the fragments. From this, after this, what is going to happen, students? I am going to transfer this to a southern blotting. Okay, I am going to do southern blotting. That means I am going to use a nylon or a nitrocellulose paper. Okay, nylon or nitrocellulose to transfer, right? Nylon or nitrocellulose to transfer, because these people are going to absorb these fragments. After that, with the help of a VNTR probe, what am I going to use? A VNTR probe, which is going to be complementary to one of the strands. Clear to all of you? Complementary to one of the strands. So if the repetitive strand is AGAC, the VNTR probe the VNTR probe will be TCTG. Okay, this is going to be the complementary VNTR probe. This VNTR probe will go and line, okay, go and line on the nitrocellulose membrane. So remember that. Let's say this is the strands, okay, now I'm randomly drawing, okay. Now this VNTR probe will go and bind if this is the repetitive strand, okay, if this is the repetitive strand. If this is the again, everyone will not have same repetitive strand, so it's going to be different, okay, repetitive strand. Now when I see it under a auto radiography, okay, because students this is radioactive, okay, when I see it under a auto radiography, I will be able to see the samples exactly. I will be able to see the strands exactly because my VNTR probe is a radioactive probe. Okay, VNTR probe is a radioactive probe. Clear to all of you? Any doubts so far? If this is clear, then your chapter is over. Okay, your chapter is over. So in a radiography, I'll be able to see the strands clearly. Okay, I'll be able to see the strands, or I can tell I'll be able to see the fragments, okay, and I will be able to detect the criminal of the crime scene. Criminal of the crime scene. Yes, under the UV rays, okay, auto radiography under the UV rays, you will be able to see where the VNTR probe has gone and aligned. So I know the repeating sequence, so I will know that this person is the criminal. Clear? Done, done, done. Done and dusted. Molecular basis of inheritance? Yes. So molecular basis of inheritance is done and dusted, students. And for those who joined late, she is coming, that is your Diwali, okay, special thing is coming, that is going to happen on Monday evening, that is technically day after tomorrow. The video is already live. All of you can go and mark your attendance, and do not fail to mark your attendance in this. All these notes will be available on Vedantu NEET English Telegram and also on my Telegram, Biology by Gopika. So I hope the session was very clear. It's a very small concept. We have learned it thoroughly, and lac operon, do not ignore, a lot of questions come from that, and also DNA fingerprinting. Okay, so done with molecular basis of inheritance. Next week Tejas series is going to happen for locomotion and movement. So as soon as you go from here, go and like the videos that is going to come further. Okay, yes, no new teacher is coming. I think all of you are happy with this team, right? Vedantu NEET English team. I don't think we need to take new teachers every 6 months, every 5 months. Okay, yes. So yes, questions will be done later, dear. I'm not mixing up NCERT line by line with questions. Questions will be done later. Okay, so thank you so much for joining. See you all in next class, and I hope all of you enjoyed the session. Do let me know in the comments how the session was, and also like the video as your Guru Lakshana to me. Okay, so see you all. Tata, bye bye. Have a nice weekend.